Hello there. Hi, I'm Ellen, and the title of today's presentation is Colocalization Analysis Prioritizes PRKCB and ELOVL4 genes for heart attack, or otherwise in aging heart. So before going into it in depth, we've actually published in 2022 this paper that which showed that 29 novel loci associates with myocardial infarction or heart attack. And in there, what we done was to actually compare between 596 MI or heart attack patients with 643 healthy patients using GWAS, and we found that actually there are 29 novel loci that are associated, which includes MUC4 and PDE1C. But from there, what we questioned was that out of these 29, which of those should be focused your attention? So what we've actually done was to do a colocalization analysis. And what colocalization analysis tries to achieve is to bridge between the genome, or the actually to be specific, a variant with a phenom. In this case, it should be an MI or the heart attack. But what we're doing is actually to use a transcriptome as an evidence or a gene expression. And the illustration of the colocalization is well shown in the figure down below over here. So in model 1, we can see that only the variant's genomic position or the variant and phenotype is well associated. But on the but the variant and gene over here, or the variant gene expression association, is not really associated significantly. Model 2, it's an opposite situation, and Model 3, we can see both of them are significantly associated, both the expression and the phenotype, the variants, I mean. But for the Model 4, it's a similar situation, but the location are far not close enough to be actually called colocalized. So there's no evidence of them being in the same LD block. So there are different variants of different effects. So let's going back to model 3, we can see on the right figure, this is a well-established figure. So we can see that the expression of STC1 affected by this variant is actually colocalized with the effect of this variant on the TC, or the total cholesterol. So what we can see is that the variant is having an effect on the total cholesterol via the STC1 gene expression. This is really just a statistical inference, but we see that on the 82% measure, we are confident. So this is a PP4 is actually the measure of colocalization. It stands for the posterior probability, and it ranges from 0 to 100 because it is a probability. And we've done the same kind of analysis in our study on the 29 novel loci and found two of them, PRKCB and ELOVL4 to be specific, being colocalized with our GWAS signal. So on the left side is the expression of PRKCB at the site of aortic aorta, which is the blood vessel, the big blood vessel, and we can see the GWAS p-value is actually colocalized with the expression p-value of PRKCB around 48%. And on the right side is the ELOVL4 V4 on the same site, and we can see the variant, a different variant now, is having a colocalization, which means that these variants, the variants over here, have an effect by these genes over here. So these genes are important, and we prioritize them for the importance in terms of the heart attack. So what are the functions then of this putative heart failure genes? And we can see PRKCB is actually involved in thrombosis of blood clot. As you can see, the blood clotting of the blood clot formation has a major impact and is causative for heart attack. And ELOVL4 is involved in the lipid metabolism. And I didn't see, I've actually hit this figure here, but if a mouse has a high fatty acids in its the heart gets overwhelmed or it has a low ventricular function, so it goes up in a heart failure. So you can see over here, it is involved in fatty acid metabolism and cardiomyocytes, and in obese mice we can see that this VLC fatty acid increase causes reduction in ventricular function. So that was it. Thank you.